meeting is being recorded.
And then we will put it on our YouTube channel, so it will be available if you want to share it with. But I also need your permission for that.
Is verbal information okay?
I think email is better, but I will send you an email.
Sure.
I just wanted to tell you beforehand, so you're aware.
Yeah, I think it's okay with like the data stuff.
Yeah, I'm not aware of anything. I'm 99.9% certain that everything you have in there is data that we've already released.
If you realize that there is something, don't worry, we can cut it.
Oh, so I think then there will be a couple of slides that I'm gonna hide.
No, Carlos, not for you. It's only for the BIO-RAD portion.
Ah, okay.
Yeah, don't worry. Because I think yours is mainly unpublished data, no?
Mm-hmm.
The work that we are doing, so yeah. I think we will include only their presentation and the discussion after.
Okay, perfect.
And is it alright if there's users from other labs to encourage them to come use the Genesis in your lab?
Excuse me?
Is it okay if like there's users from other labs to encourage them if they have any ideas to use the one in your lab?
Yeah, that's what we want.
Okay. I'm just making sure.
Yeah. The goal is to tell everybody at Cornell, potentially in New York, even in other centers, that there is this instrument that is available. We are a platform, so we use it, but it's for everybody to use.
Really, you should ask them to mount these TVs higher.
Excuse me?
You should ask them to mount these TVs higher. You mount the TVs
Higher?
Yeah. Difficult to see. Yeah. The tennis stadium. It's open. Is that what the white dome is? Yeah. I was wondering what that was. Oh. Yeah, it's US Open. Wow. Oh, yeah.
Are you a tennis fan?
No, I'm a geography fan.
Oh, okay.
Yeah.
So you just know where the stuff is.
I fly flight sims and stuff.
Oh, really?
And so like when you fly BSR over New York City, I know where you're going.
So you've got all the landmarks.
Yeah.
Nice.
Yeah. Is that what yeah, I'm big on direction, where things are located, how things look.
I had no idea.
Yeah.
So then when you take an actual flight, do you then study the landscape and compare it to what you've seen in simulator?
When I first started flying, yeah. Now, not so much. Yeah. But sometimes, especially like on approach, like when you're coming in for landing and stuff, for sure, I'll do that. Yeah. I've got like VR goggles for it.
Oh, nice. There's actually, so do you then have like the controls?
I have a joystick.
Okay.
Hopefully I can turn that on.
I would imagine so. That's super cool. Is it, was the experience the other goggles on, is it ever disorienting?
You'd be surprised.
Good.
It literally feels like someone just pulled a curtain and you're in a different world and you're fine. Yeah. Yeah, it's weird.
Which one's the you have?
Yeah.
The Oculus.
Yeah.
Version 2. Quest 2.
And I remember that actually.
So kind of a tangential topic, but something that's kind of been on my mind. I've know some people who are developing software for basically mixed reality training. So using people like the Oculus, you have in there the workflow of a protocol, say like three reference.
So just you have to like
No, I don't know.
So now I cannot hear anything from the room, huh?
Nice to meet you. Hi, Carlos. Yes, we muted until we start. Hello, can you hear us?
Yes.
Okay, thank you. Good afternoon, everybody. Thank you for joining this new webinar of the Liquid Biopsy Series, part of the HemOnc host series, hosted by the HemOnc and the England Institute for Precision Medicine here at Weill Cornell. As always, in this series, we will have first a presentation from a company representative explaining us one technology on for liquid biopsy analysis that is present in the liquid biopsy platform of Weill Cornell Medicine, and then we will have a presentation from one of our researchers explaining how we are using that technology here at Weill Cornell. So before we start, I want to remind to everybody that this meeting is recorded and that it will be published on our YouTube channel, so it will be available for everybody also outside Cornell. So I will just copy a sentence in the chat so you're all aware that even if you ask questions, they will also be recorded. All right. So I think we can start with our speaker of today, Tadas Kasputis from BIO-RAD, who will give a talk on going beyond ctDNA, explore the power of CTC analysis using the BIO-RAD genesis system.
Thank you, Carolina. It's great to be here. Thank you for hosting me and for having me. I'm very excited to be here. All righty. So outline today is I'll first introduce you very briefly to the Genesis Cell Select technology that is available here at Weill Cornell. My goal with this talk is to really inspire everyone here with some exciting science and get you all thinking about how you could apply the Genesis system in your work. And I'd be happy to, after the talk, discuss with anyone, and I'll have my email available if you'd like to discuss any of your work or applications as well. After that, I'll get into some data, some Genesis CTC capture performance metrics. And then we'll get into the bulk of the talk, which is basically highlighting various applications that we've developed at BIO-RAD, and also different research highlights, different research studies by some of the users using the Genesis system around the world. And so we'll cover about 4 sections, with Carlos going at the end to cover what's going on at Weill Cornell. So I'd like to start first with some fun bit of trivia. BIO-RAD. Does anyone know what that means, what it stands for?
No.
I was going to look in the chat, but we don't have time really. But BIO-RAD it stands for biochemicals and radiochemicals. Which is very interesting. Anyone know how old the company is? Probably,
No.
It's 73, so it's one of the oldest biotech companies in the world. And at BIO-RAD, we've dedicated over 70 years to advance our mission, which is to advance science and save lives together. Okay, and we do this by developing and commercializing various reagents, instrumentation, platforms, etc. And as part of that commitment to advancing science and saving lives, BIO-RAD has invested a great deal of time, money, and energy into the oncology space. Okay, and about 15 years ago, digital droplet PCR was introduced, and BIO-RAD is one of the global leaders in ddPCR. And ddPCR is a potent tool that is used by many oncologists for ctDNA analysis, which is non-invasive, highly specific, and cost-effective. However, there are several advantages to using CTCs over ctDNA. For example, ctCs provide comprehensive molecular profiling capabilities, including phenotypic information, and they also provide insights into tumor metastasis. ctCs are also intact, and so ctDNA could be degraded, whereas ctCs are intact and provide a multi-omic platform for interrogation. We can also use ctCs to develop novel in vitro and in vivo assays. And so ctCs, we can see, are very important alongside ctDNA and liquid biopsy research. And so BIO-RAD has recognized all that by developing, a full suite of products that are ideally suited for all facets of liquid biopsy, whether you're researching ctDNA or ctC. But for the purpose of this talk, I'll be covering the single-cell analysis systems that we have for ctCs, beginning with the Genesis cell isolation system. So we developed and optimized the Genesis system and self-select slides technology to address very specific needs that we've heard from researchers like yourselves that do ctC work. We wanted to provide a fully animated capture and enumeration device for ctC analysis that offers high capture efficiency, customizability and flexibility, increased sensitivity, increased throughput, and ability to facilitate multiple applications. So the Genesis instrument cell isolation system is in the lower left-hand corner there. It's the instrumentation for ctC enrichment and enumeration. And the self-select slide is shown in the middle there. That's the true workhorse of the system. This is the interface at which ctC capture occurs. And so the self-select technology uses size and deformability-based capture to characterize ctCs. And it does so by utilizing microfluidics, and over 140,000 of these individual microchambers, as you can see on that image on the right, located on the slide for microfluidic size-based separation. And so what's cool about these is they're perfectly suited size-wise to hold ctCs. And so this opening along the top is 30 microns wide, and then that holds the ctCs, between that range of 15 to 30 microns. And then there's a small funnel. It funnels down to 8 microns, where red and white blood cells are filtered out, and ctCs are captured on the chip. So captured cells once they're captured, they can either be stained on the slide using antibodies for phenotypic analysis, or the cells can be retrieved for downstream analysis, such as for cell culture, genomics, proteomics, approaches, etc. This platform, like I said, segregates ctCs into their own chambers while red and white blood cells are filtered out. Basically, we can process between 1 and 10 mils a sample, and we've shown very high cell capture efficiency, greater than 80% in spiking experiments. And the nice part with this system is, unlike most other ctC systems that are what we call closed systems, it just has one workflow you can use, one protocol. This has open software where we can tune parameters or even invent new workflows for your specific needs. Also, some cool images. You can see a single ctC here in green on the right, and a red blood cell, 2 red blood cells actually, that have been filtered through. And we can also have the ability to capture clusters. As you can imagine, you can have clusters, kind of appendages fitting into those chambers. Let's talk about some of those performance characteristics, such as sensitivity, efficiency, and reproducibility first. So this experiment here is a spiking experiment utilizing MCF7 breast cancer cells that were spiked into healthy donor blood and then processed using the Genesis. And so we can see here that the capture efficiency is highly linear for samples. And this does not change across the range of cell inputs, meaning this is highly efficient and also highly efficient even in the most sensitive ranges for low cell inputs, such as 10 cells, as you can see on the left-hand side there, or in the high cell ranges in the thousands of cells. We still see the same staining efficiency. Similarly here, this is another spiking study published by Wang et al. Using non-small cell lung cancer cells. And they found that when they process samples with as few as 5 ctCs total in their sample, they are able to efficiently and reproducibly capture the cells. And in this case, ctC phenotype was confirmed using a custom antibody panel that includes PD-L1 and cytokeratin, as shown in the image on the right there. These data are basically a very nice illustration on the ability of the technology to incorporate custom antibodies into the workflow and the ability to capture a range of ctCs with high sensitivity. So to further highlight the versatility, efficiency, and flexibility of cell select technology, let's take a look at this study that used different breast cancer cells, specifically MCS7 and SKBR3, which are an epithelial subtype, and the MD231 cell, which is a mesenchymal subtype. The purpose of this experiment was to show that despite the cell phenotype, the system is able to capture this. This is unbiased capture, not like when we use antibodies where we can just capture EpCAM positive cells. We can capture cells across the entire epithelial mesenchymal transition spectrum. And so they show this very elegantly by making a large antibody panel, and we can see the varying expressions for those antibodies. So staying on the topic of the EMT spectrum, this next study describes and highlights the importance of capturing and analyzing ctCs across this entire spectrum, and what that means in a clinical context, okay? So in this research, researchers from Juntendo University School of Medicine over in Japan investigated EMT status from metastatic breast cancer patients in the clinic who had received eribulin-based chemotherapeutic treatment. And so the figure on the right shows PFS as a function of time for high and low categories of enumerated cells. You can see here it has, on the bottom there, it has high and low categories, less than 2 ctCs is low, equal or greater to 2 ctCs is high. And then these are classified along the lines of epithelial. We're just classifying, or we're just interrogating epithelial ctCs, mesenchymal ctCs, or the combination, total ctCs. And not only did they find that low ctC counts correlated with improved survival, but they also found that counting the total ctC fraction has better prognostic indications, as you can see here by the p-value and the spreading of the low and high counts in terms of progression-free survival. So as I finish highlighting these several key performance characteristics of the system, this was all done using blood, which is the most common liquid biopsy medium that is interrogated. But I'd like to highlight some new exciting work that is being done by some researchers using our system using other biofluids. So for saliva liquid biopsy, we worked in collaboration with Dr. Hushan Yang's lab at Thomas Jefferson University to develop a saliva processing workflow on the Genesis system. And in this workflow, it uses 3 mL of saliva. However, that sample does require some pre-processing. So when we run blood through the Genesis system, we don't need to pre-process it. We just dilute it with PBS, throw it on the system, and it's good to go. But since saliva is, has a lot of bubbles and it's a different type of medium, we have to centrifugate and then resuspend the cell fraction and run it through the Genesis. Okay. So when they did that, they enumerated ctCs from patients with metastatic head and neck squamous cell carcinoma, and they profiled the captured ctCs using a very robust antibody panel, as seen on the top row of images there, for a captured ctC. The bottom row of images shows a captured white blood cell to show the contrast between a positive and a negative ctC. This group was also able to demonstrate that saliva is a very rich source of ctCs, as demonstrated by their ctC counts in Table 2. So you'll notice that these counts in the thousands per 3 mL of sample, in my opinion, it's high. When I typically assay 5 mL of blood, and patients will see on the order of tens to hundreds of ctCs. And so to be seeing this count in only 3 mL of saliva was very interesting finding. Next, we worked with the Sunder lab at NYU to enumerate CTCs obtained from the CSF of patients with pediatric brain cancer using the Genesis system. And so as you can see in the representative CTC images here, the Genesis and CellSelect slides are able to process CSF without any pre-processing, and they capture CTCs from CSF as evidence from the FCAM positive CD45 negative signature for CTCs. So next, we'll get into the applications and research highlights portion. This first highlight is a study that utilizes enriched CTCs for lung cancer biomarker discovery. And so while internally we conducted this research using spike-in cells as a proof of concept, but in the real world, this approach that I'm showing you could readily be applied in a clinical setting to discover and validate novel biomarkers. So in this study, A549 cancer cells were spiked into whole blood at concentrations of 50, 100, and 1,000 cells per ml and enriched on the Genesis system. Then the total RNA was purified to construct bulk RNA-seq libraries using BIO-RAD Sequoia complete stranded RNA library prep kit for bulk RNA-seq. And that was paired up with a ribodepletion kit as well. And then after that, differential gene expression analysis was conducted using specifically identified tumor-specific gene targets using QRT-PCR on a BIO-RAD Opus TSX system. So first, like I said, we use bulk RNA-seq to identify a unique molecular signature across our samples. And so the top figure shows that the Sequoia library prep kit captures the entire transcriptome, including long and short RNA biotypes. Figure A shows the abundance of transcripts that are unique to the spike and cell line. And figures B and C show the overall abundance of long and short RNA biotypes. The bottom figure shows the number of potential biomarkers unique to A549 cells that were identified across the various concentrations of spike-in. And with that analysis, we determined 759 genes that were found to be in common across all 4 spike-in conditions. So you'll also notice that we stratified this also by kind of this medium to high input of 100, 500, and 1,000 cells per mil and 50 cells per mil, because we see that less unique genes per read. So then we took those 759 transcripts identified using RNA-seq, and then we cross-referenced them to BIO-RAD lung cancer H384 panel and the prime PCR panel target cross-referencing. And after doing that, we were able to identify 34 gene candidates that you see in the table there. Okay. The figures on the bottom row show differentially expressed genes amongst our 34 gene candidates for the different concentrations of the spike-in. Okay. Upregulated genes are denoted by the red dots, whereas downregulated genes are denoted by the green. And from this data, you can see that each contrived sample shows numerous A549 specific genes that were differentially expressed. So based on that analysis, 2 genes, MET and TOP2A, were found to be significantly differentially expressed across all samples. Okay. The figure on the left shows differential expression for the cells spiked at the mid to high concentration. And in this analysis, we were able to identify 6 genes with at least 2-fold or higher expression with statistical significance. But the figure on the right shows differentially expressed genes across all 4 cell concentrations. And among those 6 genes, MET and TOP2A were found to be differentially expressed. And so this highlights the significance of these genes and their significance as consistent biomarkers for lung cancer. So BIO-RAD's Genesis and ddPCR platforms both enable molecular profiling, longitudinal monitoring, and minimal residual disease detection with high sensitivity and absolute quantification of expression. And so that's what's very unique with ddPCR compared to qRT-PCR, absolute quantification of expression. And so in this section, I will detail several studies that use both the Genesis to enrich for CTCs and ddPCR downstream for quantified PCR analysis. So for this study, as you can see in the depicted workflow, whole blood is acquired from patients or contrived with spiking cancer cells, run on the Genesis to capture the cells, and then subsequently the cells are recovered, and that enriched CTC population, we take it and we conduct nucleic acid extraction to extract DNA and or RNA, all right? Then DNA or RNA is subsequently run on BIO-RAD's ddPCR platform for SNV or gene expression analysis. And so in this experiment here, this was conducted to do some EGFR mutation profiling. So in this experiment, 3 different spike cell lines were investigated: 8549 wild type cells, and H1975 and H1650 mutated cells. These were enriched on a Genesis, collected, ddPCR was conducted after DNA extraction. Okay. This data shows that EGFR DNA mutations were detected in all mutant cell lines, and also that mutant copy number was shown to increase. So here you can see on the top line, those first 3 boxes highlight A549 cells where we only see the wild type transcripts with no mutated T79M, whereas the H1975, which has that mutation, you can see it clearly pop up there. And then when we look at the L858R mutation and for H1975 cells and the exon 19 deletion for H1650 cells, we see that the mutations are present, and also the number of copies is increasing with increasing cell concentration. So taking that approach that we kind of did as a proof of concept with spiking cells and applying it to a clinical study, this is a clinical study by our collaborators at Biodesics, located in Colorado. And here they use the same approach to capture CTCs from non-small cell lung cancer patients and profile them with ddPCR. And just so you know, we don't have any ctDNA data here, but this group is using ctDNA in addition to CTCs, and they're finding this works very well. For the purposes here, we can see that PD-L1 is detected in both early and late stage samples, and so you can see here 3 donors donated at both the early and late stage non-small cell lung cancer. And we can see here on the table below the PD-L1 to housekeeping ratio, and that they're shown to change over time. They either increase or decrease, which of course is correlated with clinical characteristics or response to therapies. So finally, this is the last section that I'll discuss before Carlos gets to it, but this is one of my favorites. It's super exciting. There's not a lot of work being done in this, but single-cell transcriptomics of enriched CTCs, which is, very new to the CTC field. So this approach we developed at BIO-RAD using our instrumentation to profile individual CTCs by single-cell RNA-seq using both the Genesis to enrich for CTCs and our new 3' dd-seq RNA-seq library prep kit, okay? This library prep kit is a droplet-based single-cell library approach, okay? So I'll get into what that means here. So in this workflow, blood is captured and enriched on the Genesis. We have to conduct white blood cell depletion after this to deplete whatever white blood cells may remain. And then single-cell RNA-seq is conducted. And so in this approach, as you can see here, right, it's kind of hard to point, but after after they undergo enrichment and they're recovered, the purified Ver cells are merged with barcoded beads and encapsulated oil droplets, as shown in the figure in the top right corner here. Okay, here you can see an enriched single CTC in green, same with cell tracker, and that's encapsulated in a droplet. Within this droplet, that's merged with the bead, this is where cell lysis and cDNA synthesis occurs and barcoding. And that's what gives us that single cell resolution. Okay. So to get to the data on the bottom of the slide there, first on the left, clustering analysis is shown. And basically what this is showing is regardless if a cell was from a high or a low spike in cell input, when we put all the data together, these cells all clustered together and didn't form separate clusters, which is good. We expected this because these are all the same cell type, demonstrated that cell input does not affect cell clustering, okay? And on the right, the figure on the right, on the bottom right-hand corner, these violin plots show that epithelial gene expression is similarly detected in both high and low spike in. We also wanted to examine and demonstrate the potential for single-cell RNA-seq to reveal insights regarding tumor heterogeneity. So we designed an experiment to evaluate the EMT status of CTCs using a cocktail of epithelial and mesenchymal CTCs. So in this case, we use, as you can see on the figure on the left, MCF7 and HeLa cells were combined and then spiked into a blood sample, enriched on the genesis, and then single-cell RNA-seq was conducted. And the figure on the top right there shows the clustering of single cells depicting single-cell heterogeneity across the EMT spectrum as two distinct clusters emerge there, the mesenchymal CTC cluster and the epithelial CTC cluster. And down at the bottom here is the analysis of gene expression and for the markers EpCAM, CARE at 18 epithelial markers, and Vimentin and EZH2 mesenchymal markers. And here you can see increased epithelial marker expression in the epithelial CTC cluster, while increased mesenchymal expression is observed in the mesenchymal CTC cluster. And what I'd like to highlight here is that unbiased selection of CTCs in this case is required to obtain this data. You couldn't obtain this with just an EpCAM-based capture. And so unbiased capture is very, very necessary for in-depth and accurate data. So staying on the topic of investigating tumor heterogeneity by profiling the single-cell EMT status, this final case highlights work conducted by Yusik Yoon's group at University of Michigan. They enriched for CTCs obtained from breast cancer liquid biopsies using Genesis system, and then ran single-cell RNA-seq. And this was able to reveal a diverse phenotype and significant heterogeneity in CTC populations. And so as you can see in the figure on the right, in a patient sample, they found considerable intrapatient heterogeneity. They detected both MET-like and EMT-like CTCs, in addition to some cancer stem cell-like populations, as well as you can see here, CSC. I think that's really cool as well. So basically, this intrapatient heterogeneity would be missed by other detection techniques, and that's the true importance of unbiased capture profiling. So to conclude, I hope I was able to show you that the Genesis cell select technology is efficient, versatile, sensitive, and customizable for your applications for the enrichment and enumeration of CTCs. Also, as I was stressing, unbiased CTC capture is crucial for elucidating tumor heterogeneity, accurate prognostication, and maximizing recovered cells. Also, CTCs provide unique insights into tumor cell biology, metastatic potential, clinical characteristics, and response therapies. Thank you.
Thank you.
And Carlos, you up?
I think maybe we can we can do a little bit of questions before if somebody. I do have some questions. Actually, also out of curiosity. So first of all, I'm very happy that we have the technology now, the liquid biopsy platform, and I'm very excited to start to work with BIO-RAD. Carlos and Nadia already worked with it, and
awesome.
they saw that the BIO-RAD technology is very performative in terms of CTCs. And like I was very curious about there was one picture, there was this slide, and I saw that like the cells were already stained there for so that it's possible to stain cell in the slide.
Yes.
Right?
Yes.
That images was very, very nice. Yeah,
yes.
This one is so nice. So yeah. So basically, when instead you are working with the RNA, you elute your secreted tumor cells in a tube, I guess, right?
Yes.
How that works.
Yep. So you can, if you want to do things downstream, like with RNA or DNA, you can enrich, so you elute the cells after they're captured. And there's automated like rinse steps too, that like rinses away all the blood, and then you capture the cells, right?
Yeah.
Or you can do the enumeration workflow, which also has an automated process, and we have like automated cartridges that have staining reagents, and those will, once the blood flows through, the reagents will engage and start dispensing reagents, incubating, and it stains it
everything in the slide.
In the slide.
Okay.
And you can take that slide off, and we have like a customized slide holder. You put the slide on it, and that's fit for most microscope stages, and you just put on a microscope stage.
And that's it. Okay. And that like the staining can be implemented with additional markers.
Yes, and so the nice part is you provide the markers, so you get to design your panels, and you get to pick what you put in there.
Okay, that's very nice. And yeah, so regarding the RNA, we said like we elute. And I was very curious about the study of the saliva. So the study of head and neck. And I was curious if somebody has tried the technology with saliva, but not like, of course, head and neck cancer, another kind of cancer.
No, but I was driving yesterday, and I was like thinking about my slides, and I was thinking like, could we use saliva for other cancers? Like, could you use it for lung cancer? I don't know. I'd be very interested to try it.
That's what I'm asking. And also like another question that I had is someone ever used it with urine?
Yeah. Urine?
Sorry.
We don't have anyone running urine right now. We've tried it. It is a little more challenging. It does require pre-processing. You'd be surprised. There's, compared to blood, it's like way less viscous and stuff. And you're like, why do you need pre-process? But when I worked with it, we found that there's proteins in the urine that like cross-link, and then they cause microfluidics to clog. So we have to first treat it with to break up disulfide bridges in the protein clots, and then you can run it through.
I see.
So we don't have anyone running it now, but we do have a workflow developed for it.
Okay. Did you notice if you are losing CTs when doing all the process to clean the urine?
When you lose them, yeah. We haven't done much work into seeing like how many cells we lose or like when we process. And you're also losing cells before processing, like when it's in the cup and there's no preservatives or anything. If there's no preservatives, you're losing cells right away.
Sorry, there is a question from Dr. Tanaka.
Hi, that was a great presentation. Thank you. I have a couple of questions. I too was very intrigued with the saliva results. Do you know if the group has done a comparison head-to-head between blood from the same patient and saliva to check the recovery of CTCs?
Yes, they have, but it's not published work.
And could you tell us, is it similar? Is saliva or blood superior?
Per mil?
Yes.
Per mil, saliva has a lot more CTCs.
Okay.
I like per mil, and this is my just kind of anecdotal estimation. Per mil, you'd have around in saliva around 500 to 700 ctCs per mil saliva, whereas per mil of blood, you'd have like somewhere between like 10 to 20.
Yeah. Yeah.
It's like a, yeah.
And is there any mechanistic basis for that?
Well, in head and neck squamous cell carcinoma, the ctCs are known to be shed into saliva.
Right.
And so this specific cancer type, it serves as a potent source. The tumor serves as a potent source of ctCs for saliva. But if the tumor was somewhere else, I don't know how potent the saliva would be for ctCs.
And would you be interested in collaborating on a study in lung cancer to do a head-to-head comparison on a few patients to see what numbers we can recover?
Yeah, definitely.
Great. We should talk of
lung cancer saliva. Yeah.
Yeah, lung cancer saliva.
Yeah.
We matching blood from the same patient at the same time.
Yeah, that'd be great. Yeah.
Okay, awesome. And one last question. In the last slides from the Young Lab at University of Michigan, where they enriched breast cancer ctCs and they performed single-cell RNA sequencing. Do you know what assay they used for single-cell RNA-seq? Was it like plate-based?
No, they used their own assay that they developed in their own lab. It's called HydroSeq.
But I assume this must be plate-based, right? Because 10x, you need 10,000 cells at least.
Yeah, plate-based. Yeah. I believe plate-based. No, HydroSeq. HydroSeq is plate-based, but not in the way.
Right. It's a different technology.
Not in the way. It's a different mechanism. But
yeah. And have they published that?
Yes, this is published right here. Whoops. So the citation's down here. I don't know if you can see it, if it's big enough, or I can just email it to you.
Yeah, no, we can see
it. Yes. Okay. What year was it published? I assume Yoon was the last author.
Yoon's last author, 2019.
Okay, awesome. Thank you so much. Great.
Yeah.
Also, Carlos had a question.
Sure.
Yes, it was related to those slides where you were showing data about the identification of EGFR mutations from cell lines that they were spiked into the blood. I was wondering if you have done something similar with a patient, and if so, if you compare to the ctDNA to see if it is like a higher or lower sensitivity.
So currently, Biodesics is doing that. Not with EGFR. Not with EGFR, though, I believe. I forget what they're profiling, but they're doing ctDNA side by side with the DNA extracted from ctCs.
Mm-hmm. But they haven't published that data yet, I guess, right?
No, they have not. And they're CRO, so I don't know if they ever will.
Gotcha.
It might be up to contracts, though.
Okay. Thank you.
Yeah.
Okay. I do have some other questions. Sorry. And regarding the digital PCR that was performed in lung cancer, right?
Yeah.
Like the next slide. I was wondering, so also for the panel of the digital PCR was performed with your technology, right?
Yes.
And about the mutation, how many mutations can you, the assay that can be designed, how many mutations can detect?
It depends on the system you're using.
Okay.
So we have like different lines of systems like QX1, QX600, QX700 continuum, and that will affect how you multiplex. Okay. And as you go up, they get more expensive and the more targets you can do.
I see.
Yeah.
And regarding Yeah, the last work you show us, the epithelial mesenchymal transition. Do you know the number of CTCs they detect in these 5 patients? Just to have an idea of how low we can go to do a single cell RNA.
So with their approach, these are all the cells, like, for this patient, okay? In the paper, I'm sure it says how many, but it's not many. Like, you can see the dots. Those are the cells. When we run it with our kit as well, like, we can do inputs at around like 1,000. And when you do CTCs and you have like depleted white blood cells and all that kind of stuff, you're around there.
That's the total number of cells that you put.
Yeah.
And doesn't matter how many CTCs you have in there.
No, we can still get data.
But do you have an estimate of how much loss you have from the genesis to like going through the other machine to do the droplets?
We do have a total, like, yeah, from when you start to spike in to like.
Because you spike 100 cells, you will lose
and after
20% of them. Then you get 80 out of your genesis. How many do you get at the end of the RNA? How many can you detect?
Isn't our efficiency on DDC 60 to 70%?
Yeah, but that's utilized niche.
But isn't that the same thing?
No. We have done that work. I just can't.
No, because we are super interested in this.
Yeah. If you're interested in that, then we'll go over all this because we have the data. I just. Too much.
Okay, yeah. No, we can stay here later.
Yeah, we'll go over.
We are very interested. We have been trying to do single cell RNA-seq on CTCs with many different technologies, and the big problem, the big limitation is the cell loss. So you need to have technologies that don't lose too many cells, because normally you don't start with. This is a big number of CTCs in this study. It's not the average. The average is between 5 and 10. 20 when you have many, so if you lose half of them.
I understand. Yeah.
Thank you. Okay, thank you. Yeah, let's move on to Carlos' presentation, and then we will have a few minutes of discussion also at the end.
Okay. Thank you very much.
Sorry, Carlos, let me introduce you. I'm sorry.
Yes.
So the next speaker is Carlos Tadas, which is a visiting fellow in medicine in the Gianna Kaku lab here at White Cornell, which is collaborating also with the Liquid Biopsy platform.
Thank you very much, Carolina. Can you see my screen?
Yes.
Okay, perfect. So thank you very much for the introduction. Today, after this talk of Tadas, I think it would be nice to provide with some examples on how are we using this platform in Weill Cornell Medicine. And we have been working with this technology, I think, over a year already. So I will give you a couple of examples on what are we doing with it. Why this is not, wait, here we go. Okay, so when we discovered that we have this platform in Weill Cornell with the Christoph and Lily lab, we decided to compare what is the recovery rate and what are the advantages of this platform with the technology that we had at the moment in the unit. And with Dr. Nadia Bayou, we set up these experiments where we were comparing Rosette Genesis, Parsortix, that is well known by the Cristofanilli team, and TellDX, that was a platform that is currently not in Weill Cornell, but we had it for a few months thanks to a collaboration that Carolina had with them, with TellBio. So with Nadia, we set this experiment. We decided to give it a test with lung cancer cell lines and also with breast cancer cells. So in this contest, it was very similar to what Tassos was describing before. We did a spiking experiment. First, we fluorescently labeled our cells with Cell Tracker, and we estimated around 200 cells per volume of spiking. And then we use 3 replicas of blood, both for lung cancer and for breast cancer cells. So the idea was to process each of these replicas in the different methods. And then we will count in the InQ side how many cells we have recovered. And by comparing with the initial input, we will assess the recovery rates for each of the technologies. So as a result, you have a summary in here. We were very happy that actually Genesis was providing us with very good recovery rates, both for lung and for breast, right after TLDX. And being Rosette-Sepp, the method that was the manual one that we were using actually was the one providing the lower amount of cells comparing to the input. So based on these results, we decided, okay, let's give it a go, these 2 platforms with real patient samples and analyze what we can get out of it. So what we did is we extracted 2 tubes of blood for each of the patients that we recruited. And one of the tubes of blood was processing the BI-RAD in Genesis and the other one for TLDX. We were processing around 8 milliliters of blood in each of the cases. And here to the right, you can see the different antibodies that we were using for the identification of CTCs. So in the case of breast cancer, we have a cocktail of anti-pan-CK, a cocktail for the different receptors that you can find on the surface of the PBMCs, and also there was HER2 antibody on top of the DAPI staining. In the case of the lung cancer protocol, we had the anti-transferrin receptor, anti-TF1 and pan-CK as positive markers, and CD45 as negative marker, along with DAPI. And then we use the microscope that we have in the unit already. So this is the table that is reflecting all the patients that they were included in this study for breast and for lung cancer, along with the different CTCs that we were isolating or enriching for each of the platforms that we used. So in the case of breast cancer, we successfully could isolate CTCs with Genesis. In this case, actually, the number was superior with TLDX, but we can say that actually TLDX was very, very difficult to use because it was giving us problems all the time. And in the case of lung cancer, we were surprised that in general, we could achieve a higher number of CTCs when we were using the Genesis platform in comparison with the TLDX. But not only that, when we were comparing in the case of lung cancer, like the type of CTCs that we were isolating, in here you can see that with Genesis, actually, you could see cells with complete integrity, and that actually we could see that could potentially be healthy if you wanted to do another type of downstream analysis like culturing, etc. In comparison with the type of cells that we were isolating with the TLDX system, where most of the cells were missing the phenotype. But in any case, this is one of the examples that we can have for lung cancer patients when comparing these 2 methodologies. And this is a high resolution picture of a beautiful metastatic CTC that we can also check after the enrichment with biogenesis. These are examples, again, for different CTCs that we could isolate from breast cancer patients, both using the Genesis system and the TLDX. So as a sum up of this part of the project, actually, we were very happy with the with Genesis in comparison with TLDX because it was giving more problems in terms of how to use it, and it was quite straightforward. Another example that we are using, now we are including this methodology in different projects involving CTCs in the Jan-Lukaku lab, and one is a collaboration that we have with Columbia where we are assessing different proteins that are associated with the metastatic state of patients. And this is a little bit like the methodology that we are using. We just get one EDTA tube of blood from the patients. We isolate PBMCs and from 1 ml of blood, and the rest of the blood we process with the Genesis BI-RAD. And then we use 2 protocols, one using pan-CK as a positive marker, and the other one is using PSMA. And then we do data analysis by using the information provided by the PBMCs of the same patient as a control. And just as an example, I'm going to show you some beautiful pictures of CTCs that we could isolate from these patients. And here you have like the classical markers for their uh, for the organoids, pan-CK and CD45 negative, when compared to PBMCs from the same patient. And in here, you can see like a clearer high-resolution picture of the CTCs expressing even different levels of the proteins that we were studying. So as a result, just to say that based on our experience, the genesis system from BIO-RAD is a very good option for enriching circulating tumor cells, at least for breast, lung, and prostate cancers. It is automated workflow, which is actually quite, I like the consistency that you can get from the results compared to other methods like Rosette-Seq that is quite variable based on the user that is performing the test. Also, as you could see, it gives you like high quality CTCs and with reasonable recovery rates in comparison with other methods, and that the quality of the isolated cells could enable downstream application like organoid formation if you would like to try, or other functional studies. So that's it. If you have any question, I'm happy to respond to any of the questions that you have.
Thank you.
Carlos.
Thank you. Yes.
That the images on the last slide are like some of the nicest images of CTCs I've ever seen. Those are great.
Thank you.
Question on the comparison of the images between the TelDX and the Genesis one. What do you think made the TelDX cells look that way? Like that non-punctate.
My bet is the high pressure that it uses for isolating the CTCs. Also, I was actually checking, I don't know, sorry, let me share again. I actually stopped sharing very fast. But interestingly, I was checking, and the patients that actually in lung cancer, I'm talking, right, that were showing a higher number of TelDX were those that they were treated naive, and those that they actually had like a previous treatment, I couldn't find any. And I think this because probably these cells are kind of more sensitive, and because of that method is using such a high pressure, probably they get compromised. That's my bet, but of course we should continue investigating further if we want to have like a clear answer of what could be the reason.
Awesome, thanks.
Any questions? Also from the Zoom, any questions for Carlos or Tadas too? I have a question going back to I don't have a question for Carlos because I know all the work. Just reconnecting to what we have been doing here with breast cancer. We have only looked at metastatic breast cancer for now. Do you have any data on early breast cancer patients?
I'm trying to think like all the customers. Not that I can think of. Might be doing it somewhere out there.
Yeah, it would be interesting to see because if there is a higher detection, maybe.
Yeah. In the early cases.
Yeah. In general, if it's detecting more CTCs, we published before that by using a outcome independent technology, we could detect more CTCs in early cases. And we think that is because maybe they are less epithelial than the one in the metastatic stage, where we didn't really see much of a difference compared to the scan-dependent technology. So I was wondering.
Yeah, no early
maybe
cases.
We would do it.
No early cases. You should do it.
And I think we are done with the time, so unless there is anything else that anybody wants to ask, we can say bye to everybody. Thank you very much, both to BIO-RAD and to Carlos for presenting.
Thank you.
We will see you next year with the next seminar.
Bye, everyone.
Bye. Thank you.